Metals are fascinating, and approximately 3/4 of all known chemical elements are metals. What comes to mind when you think of the word metal? Heavy metal music, welding. You might think of iron or lead present in soil and rocks. You may have heard that contaminated soils and mining are harmful to agriculture. In Europe, however, agriculture itself introduces potentially harmful metals to soils via fertilizers and pesticides. But do you associate metals with plants? Plants need tiny amounts of various metals to grow. And did you know that metals are also essential for our own health? Without them, we couldn't survive. Many trace elements such as copper, iron, manganese, nickel, molybdenum, and zinc are essential for all organisms. In plants, they're vital for proper functioning of proteins, including enzymes. These are required for photosynthesis and most other metabolic processes, and consequently for plant development, growth, and propagation. Unlike vitamins, plants cannot synthesize metals. Instead, they take them up from the environment. When plants don't take up enough metals, they're in trouble because of the many functions that require metals. For example, 30% of agricultural land is alkaline, and metal availability can be limited under these conditions. This may lead to harvest losses and lower nutritional value of crops. Essential metals can become toxic when plants take up too much. High concentrations inhibit plant metabolism, and the mechanism depends on the metal and its concentration. This often causes oxidative stress and alters normal plant growth. Other metals such as mercury, lead, and cadmium have no essential biological function in plants and can be toxic too. Human diets deficient in micronutrients negatively impact on our entire metabolism, thus affecting our growth, cognitive, and immune system. About 1/3 of the world's human population is at high risk of zinc or iron malnutrition, especially in developing regions with a predominance of cereals in the diet. On the other hand, some metals are toxic in high concentrations. An excess supply of trace metals may cause cancer, damage the nervous system, and cause malfunction of many vital organs. Since 2020, a network of researchers actively collaborates to investigate how plants cope with metals. This network is called PlantMetals, under the scheme Cooperation in Science and Technology of the EU-based COST Association. The network currently has more than 220 members from 42 countries, 32 of which are COST members. PlantMetals integrates stakeholders from academia, industry, and the public, and includes training of young researchers. Its results are disseminated not only via articles in scientific journals, but also via a book for children, social media, agricultural fairs, our website, and our YouTube channel. Research on metals in plants starts with a sampling campaign in the field or in greenhouses. Different plants from different environments are sampled, such as from agricultural land, aquatic systems, mining sites, mountains, trees, and controlled plant growth facilities. Researchers of the PlantMetals network collect soil samples to investigate soil fertility and soil contamination, as well as different plant organs such as leaves, roots, and plant saps to study plant metabolism. In the laboratory, a large range of techniques, equipment or analytical devices can be deployed to measure the concentrations of metals in plants. The concentration tells us if the plant is adequately nourished with metals or if it is suffering from too little or too much metals. The most important modern quantification technique is ICP-MS. Here, the sample is ionized in a plasma, and then the metal ions are separated according to their weight using electric and magnetic fields. The ions will hit a sensor, and so an electric signal is generated. The intensity of the signal generated by the atoms of a metal is proportional to the concentration of the metal in the plant sample. In this way, the total concentration in the sample can be computed. Visualization techniques such as microscopic imaging of X-ray fluorescence, so-called micro-XRF, are used to localize metals in plant tissues and cells. This instrument uses X-ray light to see the metals. The localization of a metal is often directly related to how the plant copes with metal stress. Seeing the chemical partners of the metal also provides crucial information. For this, we mostly use synchrotron X-ray absorption spectroscopy and LC-ICP-MS. In daily life, sometimes we may be able to differentiate with the naked eye what a metal binds to. For example, we can distinguish pure iron from iron which has partnered with an oxygen atom, that is, rust. The measurement of X-ray absorption works on a similar principle to determine whether a metal is alone or interacting with surrounding atoms, for example, from proteins. To identify such proteins, a liquid chromatography unit is coupled to an ICP-MS machine. This helps in separating the candidate compounds, and we can figure out to which proteins the metals are bound to. All this information about the metal then helps us to predict what purpose the presence of metal might serve in the plant. To learn how the lack or excess of trace metals affects the metabolism of plants, we need to measure key steps of this metabolism. A powerful tool for this task is to study the changes of light that are produced by plants as so-called fluorescence, and this can be done in an imaging way to find out where in a leaf damage has occurred. Further, we measure the release and uptake of carbon dioxide by plants. This is a gas that you may remember from global warming. These measurements allow us to analyze how much carbon dioxide plants eat or breathe out depending on their metabolic state, and how this is regulated by little openings in the leaves. We use genetic approaches such as mRNA quantification by PCR techniques. These help identifying genes encoding proteins that are responsible for the transport of trace metals into the roots, their distribution to other plant organs, and their use in the metabolism. Using these techniques, we can establish how plants manage to ensure an adequate metal supply in a changing environment. The collaborative research of the PlantMetals network aims to better understand how plants take up and use metals, and how they cope with variations in metal supply, that is, too little versus too much. This will contribute to a more effective management of the effects of metals in the environment and agriculture. It can furthermore be used for improved risk assessment of pollution and for better and more environmentally friendly control of plant diseases. Ultimately, it can lead to growing more nutritious and healthier crops and the decontamination of soils by plants.